I welcome you all to today's session of Mr. Manish Yadav. Mr. Manish Yadav has done MS in Analytical Instrumentation from MP in 1998, presently pursuing his PhD in Bioanalytical Chemistry. He has more than 22 years of experience in a biopharma R&D, bioavailability, bioanalysis, GXP, QMS, audits, and trainings. He is CEO and founder of Ethics in Consulting and Result Solution Ahmedabad. Also WHO trainer BABE trials and inspections, Geneva, Switzerland. He has experience of a train and trainer, Ministry of Skill Development and Entrepreneurship, India. Authored 50 plus research articles published in a reputed international journals. Delivered 50 plus invited talk and national and international conference. Established and successfully run 2 CROs, 3 BABE centers and 9 bioanalytical labs. As auditor, successfully conducted 300 plus third-party audits and monitoring. Trained 1,000 plus bioanalytical clinical BABE, QA, GCP inspectors and regulators globally. Executed the first HPLC MS and MS experiment of bioanalytical lab of Aurobindo Bin, GVK Bio and Vida CR. Worked for a reputed companies like Lambda, Toronto, Cadila and many more. Thank you.
So let me start my sharing my screen. Okay guys, so dear friends, myself is Manish Yadav, and today I am pleased to speak on behalf of Gujarat University. When they requested me, they invited me. I am honored for this opportunity to present, to talk to you. Basically today I want to talk on the Green Lab online refreshers course in chemistry, so organized by Gujarat University. So today I'm going to talk on Green Lab, its sustainability and future perspective. So myself, Manish Yadav, and I am founder and CEO of Ethics right now. It's basically a consulting firm. And I'm WHO trainer from APAC region, appointed through Geneva, Switzerland. I'm the trainer also. So with this brief introduction, let me start what I want to talk about. So I would like to talk or start my talk with a philosophy which I follow in my life. It's through George Bernard Shaw, and I strongly believe in this, which I followed throughout my life in pharma career when I worked for 16 years. And after that, I decided to leave the job when I was associate vice president at Alchem Labs, Mumbai. And then I started my own company named as Ethics CRS, based out of Ahmedabad. So I strongly believe in this philosophy where I believe if you have an apple and if you have an apple and I have one apple and we exchange apples, then you and I will still each have one apple. But if you have an idea and I have an idea, and if we exchange ideas, then each one of us will have 2 ideas. Thank you so much. Now, with this is the outline of my presentation. So part 1 will be around, I believe, 40 to 45 minutes. There we'll discuss 2 chapters, understanding of bioanalysis, bioanalytical lab, and its dynamism, basically the progress. The second part will be covering the current challenges for current labs, existing labs, and if somebody wants to come up with a new green lab, new concept. The second part will be covering understanding of expectations on green lab and future perspective of green lab. Let's start with part 1. Understanding the bioanalysis, bioanalytical lab, and its dynamism. So let's spend some time to understand, if we want to understand bioanalysis, if we want to understand the progressiveness, the nature of bioanalysis, which is highly progressive and dynamic, we need to understand what is the science, what is the science behind it, what is basically bioanalysis. So analysis of drugs in biological matrix, that is what is bioanalysis. If I talk about bioanalysis or bioanalytical lab, it's an integral part of drug development process. Whether it is, so drug development is a big cycle. It will take almost 12 to 15 years. Earlier, it used to take maybe 20 to 30 years to develop a new drug. So if you want to develop a new drug, I'm basically from pharma research, so I understand. Nowadays, it is reduced to maybe half of the time. So it will take, still it will take maybe 12 to 15 years. Now you can relate this thing, what I'm talking about today's scenario where we are facing a COVID-19 virus challenge. So what I say, basically, people are talking about vaccine will come from day one, means probably from March. I'm hearing it will come in 3 months, it will come in 6 months. Somebody was saying no, it will not come before 2 years, because the logical reason which I understood, my PhD is also on antiretroviral. Antiretroviral is mRNA basically. And when we talk about COVID-19, it is also mRNA virus. When we couldn't find the vaccine for mRNA virus, which is existing from last 30 years, how it is possible to find even a good vaccine for a new virus, which just came in existence within even 3 years, if we get, it is a big achievement. Proper vaccine I'm talking about. So drug development process is a big process, lengthy process. It is time taking because we have to see not only its safety, the effectiveness of that drug. So bioanalysis, anywhere when we talk about PK studies to support drug development programs, bioanalysis is an integral part. It's an integral part of drug development process, any process, whether it's the INDA, investigational new drug application, or abbreviated new drug application, which is ANDA. A regulated bioanalysis, basically when I talk about, comprises of 3 vital components. It's an amalgam of basic science, which is analytical science, you can say, talk about chemistry, analytical science. Then quality aspects, quality practices, I say, when you talk about GCP, GLP and all those things. Then regulatory considerations. So which country or market you are targeting to launch your drug. So that country's rules of the land, the regulations will be important. And all 3 things I should keep in my mind before I develop and launch this drug to particular market. So bioanalysis will cover all these 3. So somebody has to have information and knowledge of these 3 areas. But over and above all of this, ethics is something which is focal point on all the fronts when I talk about bioanalysis. So ethics we'll talk in between so many times. So ethics is nothing which I believe when we are, or I am working alone, when nobody is watching me, how I am working, what kind of policies, what kind of SOP, standard operating procedure, or what kind of intent I am keeping, whether I want to finish my job like that only with the quality, or fatafat, fast, or it is with proper way. All these things made a character, and that character is important. So ethics is something basically we learn from our parents. They always teach us do the good things in a right way. So that is what ethics basically. So do the things in a right way, whether somebody is watching or not, that is all. So ethics are something, it is very important point when we talk about regulated bioanalysis. Next is when bioanalysis is there and you want to carry out, you have to have proper setup, and that is called bioanalytical lab. So what is the principal goals of a bioanalytical lab, ideal or typical bioanalytical lab? So bioanalytical lab is basically intended to develop a high quality bioanalytical methods with the characteristic of what? Basically error resistant, less downtime, less rescheduling. Means when you use this method, we should have proper identified criteria. All the values should be within that criteria, well within that criteria, not at the border even. while validating the method after development. So well within the criteria. When we apply this method after validation, this method should not have much error. So it should be error resistant, should not fail again and again. We run a lot of samples in batches. So those batches failure rates should be nothing actually. It should be zero actually, ideally. But even then, if there are failures, should be less. So downtime will be less. Otherwise, we will keep wandering, spending time in troubleshooting, and there will not be any performance. So again, rescheduling of batches, analysis, reanalysis. We'll talk later on this part of the, maybe next part of this presentation. I'll try to talk on repeat analysis and other things. So rescheduling. Another important aspect, it should be cost effective. We cannot, when we talk about regulated bioanalysis in commercial space and anywhere in the world, the good bioanalytical lab has to have some performance criteria. They have to perform. They have to be profit making. Otherwise, nobody will run that facility. So there will not be any existence of good bioanalytical lab. So it should be profitable. So cost effectiveness is also important, but not on the quality of cost of quality. Right? So quality has to be there in all aspects. Generate lab should be able to, or method should be able to generate reliable, reproducible testing data. And here it comes to data integrity, which is a very high focus area for today's regulatory agency. On any auditor or inspectors coming to your facility, they see this aspect critically. So data integrity, we'll talk again later. For a bioanalytical lab, method development, when I talk about, is a core activity. We need to develop a lot of methods. So it's a continuous process. Quality methods we need to develop within a stipulated time. It demands extra efforts along with management of other time-bound routine projects. So when somebody is working like a senior scientist in bioanalytical lab, he has to not only continuously focus on development part of methods, various methods, one or two, whatever his responsibility within that group. He has to do other routine activities like checking the data of existing analysis, other projects, guiding, training the newcomers, sorry, developing the team and others. With that, they have to focus highly on development part because method is core. If method is good, their life will be smooth. If method is not good in a longer run, they are spending a lot of time in making up that method, rescheduling of the things. So it is time consuming and definitely not at all cost effective. When I talk about other important things, how will we decide whether this method is good, what we are developing, or how we develop a good method? What are the characteristics of this method? So high quality method is in high demand. And I always tell to my team, high quality methods, it will be in demand, always be, because it was, it is, and it will be. And I relate this thing with one of the example there. I say, good people always good people always are in demand, because even a bad person needs a good person. Similarly, high quality method may be a little bit taking more time. Sometimes we feel sticky. Sometimes management come and pressurize us for timelines and other things, but we should give enough time to develop this method. To have quality output, we have to spend time in development so that later on we should be able to save time, and it will be in multiple. Because if something happens after development, after validation in analysis phase, it is really a wastage of time. It's a huge loss. And sometimes you lose even clients and businesses because they will not come back if you are not able to deliver quality results in time. Anyways, so the quality of bioanalytical method can be decided by 6 M's. This you all should remember. This is very important slide. The 6 M's basically are man, machine, methods, material, milieu means environment, and management. So quality of any method, analytical method or bioanalytical method, will be decided based on this. And we should focus on these sub criteria also while developing the method. Like when somebody is trying to develop a method, we have to have skilled manpower. We have to have skilled manpower. Machine has to be calibrated, robust and qualified. Only then we can use this machine. So I'm saying without checking this, if we started developing and we developed and validated also, then later on the auditors will come, they will never accept this method because they will check this aspect. Methods, whatever methods, preparation methods, solution preparing, mobile phase preparation, biological sample treatment, these methods has to be properly characterized. Again, it's a part of development, then we validate those processes also. Should be suitable and documented properly. In regulated bioanalysis, I say, where I'm saying reg bio, it's a regulated bioanalysis. Short form is reg bio. Here, the science is very important, as you all know, in analytical science. So science is important, but equally documentation is very important. If we don't know how to document my experiments, we are not the people who can sustain in bioanalysis, regulated bioanalysis. Another aspect is material. So what are standards or material or liquids or solvents we are using should be of proper quality. They should be with COA. Even the, suppose we are using the drugs, the molecules which we are dissolving in the solvents, making solutions, those drugs also should be properly identified. Then milieu means environment. We have to have proper temperature controlled. Most of the labs are AC, air conditioned, centralized air conditioned. These labs and everything should be properly maintained. Two things, especially temperature and humidity, and irradiations, radiations, quality of radiations, vibrations. So like if I take an example of analytical balance, should be vibration. We should put these analytical balances on vibration free tables. Last is management. So all kind of support, all these materials, manpower, skilled manpower, good machines, okay. Management is responsible to provide all these resources. Only then an analyst or maybe group leader will come and probably develop this method. So method, when we are developing, we have to keep these 6 M's in our mind. This is very important slide. And a good method can be achieved through extensive testing knowledge, time and efforts. So many times we need to do testing. Once we find results are good, we need to do further testing. We need to we need to put variations in conditions little bit here and there, like pH. If there is a pH of 3 we are working, then 3.2, 2.9, 2.9, 3.1, how it is working. These kind of variations we have to do, and then we have to clear my method is still robust enough. In a longer run, method has to be cost effective. Now longer run I'm talking about. If you put good time initially in the development, you will be having less downtime and rescheduling in later phase. Highly desirable for management front, multitasking job. So when people are doing multitasking job, the good support from management is important. Next is, I'm coming to the lab infrastructure. There's a standard lab. You can see this is sample processing area. When I talk about bioanalysis, regulated bioanalysis, it's a part of clinical pharmacology, most of the time, the PK studies, pharmacokinetic studies. So when you talk about pharmacokinetic studies, especially bioequivalence, BAB, we say BAB studies, bioavailability, bioequivalence studies will have two parts. One part, clinical research, another part, bioanalytical research or bioanalytical lab. So clinical research, they conduct the study, they call the volunteers, they follow rules, they follow GCP, they follow good clinical practice, they follow their SOPs, protocols, study specific protocols, procedures, and they call the volunteers. Volunteers will stay, they collect the samples as per schedule and design, study design, they collect the samples. They store the samples, their time being, and then they transfer the samples. Depends on, again, requirement of the drug, stability and all of those aspects. Then they transfer the samples to lab, bioanalytical lab, another part of that study. So it's a bioanalytical research, bioanalytical lab, their samples will come. So our job, bioanalytical people's job is to analyze drug available in those clinical or human samples. So for that, you need a lab, sophisticated advanced lab. I'm saying you need very good air conditioned, temperature controlled labs because most of the pharmaceutical drug components, they are unstable at high temperature. So we have to control our analysis. So we need controlled environments, not only just to show, it is to follow. To get a quality method, we need quality controlled or well controlled lab. So we have in lab, we have two parts. First part, a chemical treatment area, a sample processing area. Another part, equipment wing, where we have equipment. So you can see here, we have equipment. And in typical bioanalytical setup and today's recent or current bioanalytical lab, most of the labs, 99%, they are using only one machine. The platform is only, analytical platform is only known as mass spectrometer. And in today's era, we use tandem mass spectrometry, like maybe triple quadrupole is most suitable machine. Triple quadrupole mass spectrometer is the most suitable detector in pharmaceutical, or I say even the bioanalytical or bioanalysis, regulated bioanalysis. So it's basically a machine is LC-MS-MS. So it's HPLC coupled with mass spectrometry, tandem mass spectrometry. So that is what I say LC-MS. It's a standard lab which I created along with other guys, and this lab is running very nicely. Not only having 14, now they have 49 LC-MS. They are global. This lab is really very successful and big, located in Ahmedabad and across globe, so many units. So so a standard lab will have a processing area, equipment wing. Then in equipment wing, we will be having proper setup for equipments, all calibrated, validated, identified, properly maintained machines with the documentation and everything. Here we have all the support system, chemicals, reagents, mask, fume mode, everything available to do the routine job, whether it is development or analysis. We have to have a centralized temperature monitoring system. Standard is zero therm. For deep freezers and other freezing devices like refrigerators, we put our working standards, reference standards for analysis, for making solutions, and then analyze those drugs. Watson LIMS, it's a LIMS. We'll talk again in second part. What is green about that, right? So the next is, okay. So it's a typical bioanalytical setup. So typical bioanalytical setup. There, I talk about, I say, everything starts from HOD, so head lab operations. I say then he's having a lot of hands. There maybe in charge of the lab will be the second authority, like senior principal scientist, then group leaders, then senior group leaders, then group leaders, bench level people like analysts and lab supporting staff. Here we say custodians, maybe washing people are doing, those guys, maybe undergraduate and that kind of qualification we need. But from analyst onwards, I say the postgraduates are being preferred. Then maybe equipment engineer is on site, or maybe they outsource this activity. But on site is ideal when we have more machines. Watson LIMS, it's a laboratory information system. We'll talk later on this. Then report writers, custodians, as I mentioned, then bioanalytical quality monitors. So quality monitors, in lab, we say quality control, like plants and others. In our labs, like when we have bioanalytical labs, bioanalysis we are doing, we say bioanalytical quality monitors. They are reporting to head HOD of the lab. And otherwise we have QA also, CQA, QA, like centralized QA, quality assurance or quality assurance. They will do at the organization level, all the quality checks. These guys will do quality checks, review and everything within the department. Like for clinical, they have their own CQM, clinical quality monitors. For lab, we have bioanalytical quality monitors. Right, so, and ideally it was very old data, 92. Now in the same lab where I talked about, located in Ahmedabad, we have more than 500 people. So now coming to the important aspect, when somebody is going for development, what is the starting point? So I'm just covering quickly. Like they have to have proper literature search. So these are the few references. I'm not going through reading each and everything. You can refer even later on. But these are the standard research articles, what we follow, what we read and follow. So these are standard reads recommended, like EMA guidance, strategies for matrix effect, which is very important criteria to qualify a method. Then you have LCMS-MS methodology, HPLC-MS-MS methodology. Brazilian guideline is also considered a standard, but the most important standard, which is, I say, gold standard to follow even to develop a method, the criteria what we draw from the guidance is US FDA guidance. US FDA guidance for industry for bioanalytical method validation. The first guidance came in 2001, and second draft came in 2013. Then again, final guidance came in 2018, and then some other things. So we have also some good reference for the guys who wants to develop a good method. So what we are targeting when we are developing a method in bioanalytical lab, especially in regulated bioanalysis area, we need to have a good reproducibility of that method. So method has to be reproducible, method has to be robust, robust method finalization and testing protocol. So robustness, how you check? You have to have proper testing protocol. I'll give you a glimpse for example how we do that. And method ultimately has to be fit for the purpose. It should not be very hi-fi method like for research publication purpose we are doing, because we have to perform within the time frame. That method somebody will award, outsource. So if I talk about contract research organizations, CROs, they get the business from pharma research companies where they are not able to manage the outflow, the basically overflow of their work. They give outsource to other companies, third party, which are good and cost effective. So they give there. Basically the quality also has to be there. So they expect very good quality methods because somebody is paying, they expect, and within time. If you are not able to give the results and the desired data reports on time, it is useless for them. So method has to be fit for the purpose, good quality and within the time frame. It's a typical bioanalytical workflow I am just trying to explain you. So how it is? If I talk about method development, so bioanalysis basically having three parts. So first thing, method development. Second thing, method validation. And third is bioanalysis, analysis, basically analysis of subject samples or samples which you are getting in lab. So first part is method development. So everything start from collecting the information, literature search, as I shown the references, and then we make solutions. So we need to weigh that particular component or drug material. So we weigh and then we make solutions. Next is we prepare samples. So how you prepare sample in bioanalysis? So bioanalysis is to be done in blood. So blood is having two components basically, red blood cells, red color if you see of blood, it is because of red blood cells majorly. And then we have other cells also, but then majorly red blood cells. And second is watery part, that is called plasma. In pharma industry, 90% or 95% bioanalysis will be done using plasma component of the blood. So here if I say sample preparation, it means we are adding particular drug in plasma. So this is called fortified samples, whether it is standard calibration to prepare calibration curve. Against that we'll analyze the QC, and unknown samples we'll get the concentration. So whether we are preparing a standard curve, a standard calibration curve standards, or quality control samples, we need to add particular drug concentration, so that we'll get the final concentration of that standard. So like 1,000, 500, 250 nanograms if I talk about. So we add accordingly drug, maybe less, very less, 2 to 5% of drug solution in plasma, because it should not having more component of aqueous sample, what you are adding from outside in plasma. Otherwise it may change the nature of plasma, which we don't want. So it should not, because actually we are getting drug in plasma from the samples we are getting from clinical human volunteer. When they are taking drug, then we are collecting samples. So it is naturally happening. So in that we have very small amount of drug, and that is pure plasma we are getting from clinical research department as a sample, where we need to identify or quantify the drug. Here we are fortifying, we are adding little bit sample solution in plasma and making those standards. So we are making standards quality control samples for analysis. Sample prep, then we do treatment. So treatment basically, majorly we are adopting three methods to treat samples in bioanalytical lab or bioanalysis. First is protein precipitation, where we just add the acid or methanol or acetonitrile, so that this plasma with the drug, basically we mix it. So we make it as a standard, then we add this acetonitrile or acid. It will suspend the solid part. Like plasma is having a lot of protein, fats, so it will suspend all those things. We get supernatant solution. That solution can be simply taken out and injected on the machine, whether it is HPLC or HPLC MSMS. So this is the simplest way of doing the sample prep or chemical treatment. So protein precipitation, so protein, fats, everything will suspend it. Supernatant can be taken out after centrifugation. So you mix the acid, then you centrifuge, and then it will be a clear supernatant liquid. You can take it out. LLE, it's a very old established process where you can have liquid extraction. You add organic solvents based on, again, science, understanding the characteristic of the drug molecule, and then you decide which kind of methodology, solvent system or solvent you add. In plasma, you rotate for 20, 30 minutes, or you vortex 100 samples like this, 100 samples all together, and you get the, again, there also you get a lot of mixing, but then we centrifuge. These tubes are 5 ml or plastic polypropylene tubes. We centrifuge, you again get supernatant clear organic layer that can be transferred to the another new tube, and then we dry it, and after that we what we say, reconstitute in mobile phase, and we inject on. Then there is another, this is liquid. There is a better methodology, we get more clearer sample. Then the final best methodology is solid phase extraction. Here it's a kind of small columns, you can say, C18 material, another mixing of material you can do based on nature of the drug, and then you select these SP cartridges, solid phase extraction cartridges. There we have loading of samples, like first couple conditioning with water, methanol water, then loading of sample like plasma sample, again as I mentioned, standard quality control and samples you load. Then you apply the pressure vacuum, and this will go off. Drug will be retained in these columns, small columns, cartridges, and then you wash it with other buffers and other like mix of solvents. You wash the bad part, dirty part of the sample, and you get your drug should be retained. So you select sample solutions like that. Okay, then finally you break that bond through a very strong, like maybe acetonitrile, or you use methanol with acidic condition. So it will break the bond with the partial, temporary bond of drug with cartridge material what they had. So you can get the pure drug in liquid, that, and that liquid can be again dried and reconstituted with the mobile base, and you can do that. So it's a long process. I will not go into the method development, but I'm just trying to tell you. So this is a standard flow. Once you select which method is better based on, again, results and combination trial, but these trials has to be done scientifically based. So we are getting results, based on results, you change your methodology approach, and then you go ahead. Like that, you probably finalize SP. So your processing method will be solid phase extraction, and then you go for another part, MS, HPLC, like you have HPLC, MS. So your MS, you have to tune, you have to optimize for the molecule. So start getting spectra, you start getting peaks, you optimize LC condition, there you have mobile phase, you have columns, everything. So it's another machine which I can't put the picture, HPLC. So you have three components here, basically. So method development will have sample preparation methodology, HPLC optimization, and optimization of MS. So you optimize MS with the parent and daughter ions, and then you set everything is ready. Okay, there also mobile phase selection, again, a lot of chemistry, pH. Based on that, you select your columns. Column chemistry, you should know. You know solvent chemistry, you know mass spectrometer, so you know physics also a bit with the chemistry. Once you are good in this, you can develop, optimize these conditions, and then finally you get the bioanalytical method. It's a bit complicated. It's not typical analytical. Because of this sample prep area and combination of two machines, it has become complex area. Sample prep itself is a different, because it is not only chemistry. You need to have a lot of biological biology knowledge, because we are playing with the human samples, which is again blood, plasma, urine, serum, all these things. So finally, we optimize the method and we say my method is ready. Then we'll go for a small experimentation. Before validation, validation is a big experiment, small testing experimentation, right? So these are the experiments. We do precision accuracy batches, we do recovery experiments, we do specificity like selectivity, whether my method is selective or not. We do matrix effect. This is again related to selectivity, but more intense methodology. And then wet extract means in liquid state my drug is stable. Then we again matrix effects. Another part is phospholipids. We check different masses. In presence of these masses, how my method is working. There should not be any interference or suppression, like some signal enhancement or signal suppression because of the presence of anything. And finally, stability studies, a lot of like benchtop stability, long term. In deep freezer, we put at 29, minus 20, minus 80, and then we put for hours, we put for days. So we check long-term stability, we check short-term stability in solution, in plasma, in clean samples. So a lot of things we need to do. But these all small version, like some 2, 3 samples, 3 samples, and less overall batches. In validation, we have proper guidance, how many samples, how many batches you need to do. So those minimum experiments you do. But it's still a good method development person, if somebody wants to develop a method, will take 8 to 30 days based on molecule criticality, molecules complex nature, and knowledge of the person. This is a typical testing protocol. I think if you are not able to understand all these things, it's okay. Because it's a high level protocol for bioanalytical people who are doing in routine. So as I mentioned, you might have heard these terms, but that's fine. Linearity, precision, accuracy, sensitivity, equipment. We do reproducibility check using different equipment, different column, different analyst, and ISR, and then matrix effect evaluation, right? Specificity, ion suppression, post-column infusion, all these selectivity, crosstalk. Two molecules are not interfering with each other, so quantitation is okay even in presence of other molecules. All these things we do, because in human body we can take other drugs also in presence of one drug. So all these things we anticipate, we get the literature, we test in lab, and then we say clinical guys, okay, these things are okay, we can take care, you just give the samples, we'll evaluate. Stock solution stability, benchtop stability, freeze thaw, like we freeze, then we thaw. Remove the sample, put on bench, put for some 1 hour, then again you vortex process, and then again you freeze, again you do thaw. As a 3-4 times we do. So 0 hour, fresh samples we analyze, then we analyze third cycle. So against that we compare. So a lot of good things, interesting aspects. Some other new experiments we need to do, like whole blood stability, interconversion, analyte reactivity, concomitant medication. Like this information we'll get from the clinic. Based on that, we decide our experiments and do that. Batch size, more samples, 100 samples, 200 samples. Now coming to the method validation. Very important aspect, as I mentioned, US FDA, EMA. EMA is European guidance, US FDA is US guidance. These two standards are gold standard. But if you talk about one, then I say US FDA method validation guidance is gold standard for bioanalytical people. So you follow that, we follow that. It is 2018, we have the latest guidance we received. and that everybody is following across the globe. So for us, whether we are sitting in Ahmedabad or Hyderabad or Delhi or New York or London, somebody is validating bioanalytical method will follow single standard across the globe. This is USFDA. So it's a gold standard, which I learned. So I did all my publications, more than 50 plus publications from India. While sitting in India, working in India, we did that, international publications. So this work is acceptable because we did as per those standards, whatever we have done. So these are few things. Again, I'm saying some good references. But a bird thing for our methods, validating any method, we apply these two standards, basically mix of these, but primarily USFDA, which is again considered gold standard, and all methods developed using specific biological matrix. Like we cannot change the matrix while study. Once we validate the method, then we inform them, and then say, no, in place of plasma, we are giving you serum. That is wrong. So we cannot analyze samples if I am validating method for plasma. So either I have to validate, redevelop and validate the method for serum, then only I analyze serum. So why to do that? So be informed, and then accordingly things will happen. There's a repetition again, how we do that. So it's a method validation. What we do, once method is set, we have SOP, method SOP in place. We train the guys on that. We delegate the guys, what are different analysts, how many, group leader, this person, that person, we do this. So delegation of responsibility to the people, and then training. So again, somebody will go and weigh the compound, make the solutions nicely, correctly, in presence of QA. So quality assurance QM guys will do in process. I'll come to that point. Then we do sample because method is set. So method is set, right? And everything is set. Sample preparation, I say sample preparation is done according to method SOP. Then these experiments, these many experiments we need to do that. So we do everything, then whatever data is being generated, data compilation somebody is doing, report generation is happening, and then group leader will check by 100%, and then bioanalytical quality monitoring people will do 100% check, QA will do 100%. In some cases, QA will do 50% or 30%, maybe 10% based on their SOP. So validation is a regulated area, understand. When somebody is coming from outside agency like USFDA or European agency to audit our facility, this document will be checked against the guidance available, whether we followed those processes. Our SOPs are designed based on those guidance. So, and report and everything will be there. So QA report will be there. So they start with report, then they'll check the data, they will check the machine, everything is okay. They will check the preparation forms, what we record. So everything will check against the report, then a report against the regulatory agency guidance. All these things will happen. Standard validation will take 5 to 10 days. Now there are some other areas like deep freezer custodian. Deep freezer, so deep freezer custodian will do a lot of good things. They issue the samples, electronically it is happening. And sometimes deep freezer custodians will have multiple job responsibilities, like column custody also in their custody, columns and plasma and standards. So the single guy or maybe 2-3 custodians team will handle these 3 things. So, but standard practice, they are more responsible to manage these samples. So samples are coming from outside, they take inside, they document it, store it properly, documentation will be done. Then whenever there is a need, they even issue these samples to the lab. The analyst will do as per request, processing and everything they do. And then they return these samples as soon as they thaw and they take these samples. They return these samples, which will go inside the deep freezer. And then, these samples, once they treat the samples properly, will go to the machine, and then records will be to the checking and then finally archiving. But all these things can be done like this. The deep freezer sample, when we are storing, managing, we have to have temperature monitoring device 24/7. It should record. all devices like 1, 2, 3, 10 devices across the facility, continuously there should be temperature recording. And we should have logbooks. So whenever sample coming inside, going outside, they should record it. Now, another important quality department in lab is biomedical quality monitoring. They monitor everything. Like they monitor, review all kind of data. They do in-process audit of sample preparation. They do in-process audit of weighing the compounds, machines, calibration standard, deep freezer standard, how they are maintained, and then processing, how they are processing as per SOP. Okay, so QA, they inform to HOD, they inform to QA also. And then they do a lot of preparation of maintenance and calibration schedule and maintaining those things, reminding people on time. So BQM is a core activity in biomedical lab to maintain the quality. Now, I talked about basics of bioanalysis and a typical biomedical lab. Now, why I'm talking about all these things? Because I want to talk about green lab. What is going to be the future lab? But I feel one thing which is important to understand before I proceed for that part is to understand bioanalysis is a dynamic field, dynamic area. Basically, regulated bioanalysis is highly dynamic area. Why it is happening? What are the reasons? Let's discuss that. So, dynamism. From last 2 decades, if I talk about bioanalysis, like I started my career in 1998, so it is now more than 22 years. I have seen tremendous advancement in scientific area. Earlier, we have very few literature available online. Now, if you go in Google and search LCMSMS, you find lakhs of articles and papers and publications, right? A lot of things, white papers, conference reports. I typed in 2000, I remember 2001, 2002, I was in Hyderabad, I was there. I typed LCMSMS in Google. I didn't find single file. So that's a way of advancement happened. Now, what happened with that 2 decades basically, better scientific clarity, understanding of allied sciences. Like bioanalysis is not a single science. It is an amalgam of 2-3 sciences. It's a chemistry, it's physics, it's clinical, biological, and formulation, because formulation department is basically formulating the drug, which we are going to dose to the subjects or volunteers in clinic, and we are getting samples, analyzing. So I need to have probably this information also. When there is a trouble, there is a challenge. If we are having this knowledge, will be helpful to me to investigate challenge and find the solution. So advancement now, even in the allied sciences like clinical and formulation, we correlate and do the things. Extended experimentation for quality and reliability. So initially we used to do precision, accuracy, selectivity, few experiments and say method is validated. But now we do lot of extensive experimentation. I'll take it in next few slides. What type of experiments and experimentation added now with the standard one. Good quality practices. Earlier we even hardly knows what is GLP. Just common sense we used to utilize and used to do that. But now we have a lot of good quality practices available. GLP was long back came, but people realized and started understanding last decade only. So, but in bioanalysis, not only GLP, we'll talk about the quality standard also. We need to have other good practices also, because it's a mixed hybrid science. So for hybrid science, we need hybrid standard practice. Regulatory advancements. So as you can see, few of my earlier slides, but here also, a lot of audits happen to Asia Pacific, India, especially India grown very nicely in this area. In 1998, we were nothing. And by 2010, 11, we were probably among top few countries, states in world, like after America and Europe. Along with them, we are considered very good in bioanalysis actually, especially regulated bioanalysis. So a lot of inspection, because we did a lot of work for US FDA and Europe. So a lot of agency inspections triggered. They came and they audited and they find, obviously, people do good or bad. When do work, mistake may happen. So they find a lot of gaps. They issued warning letters. We were lucky, we learned things. We used common sense. We did extra. When we were confused, we did extra. Some people, when they confused, they didn't do, so they got caught. But we were okay, lucky in some cases. So I'll not praise myself, but I say there are few guys who did very good job, even that confused area. But now things are very clear actually because of these advancements. So what happened? A lot of inspections happened, a lot of gaps, findings, observations came to people, a lot of failures identified during inspections, which were again considered as crime. So they issued warning letters, they issued 483 means that's a kind of notice of concern from WHO, okay? So what happened? All these things made us to understand we need to learn what? It leads to transformation in regulations and quality aspects. So we learned not only science, we learned regulations, what kind of regulations they are growing, implementing in their countries, especially US FDA, US and Europe, and then quality aspects. Because most of the Indian companies, they were launching their drugs in US and the European markets, because those are lucrative markets. There you have very good money. And with quality, if you launch, if you use your drug, you will get very good clients. They can spend money for their health. So that is the reason a lot of audits came from those reasons, and we learned. But out of this outcome, we learned a lot. This leads to transformation in regulation and quality aspects. And other important aspect, advancement of new bioanalytical method validation guidance came into picture. So many, if you see last 10 years, EMA, which is Brazilian, then ANVISA, EMA is Europe, ANVISA is Brazilian, US FDA 2018. And I mentioned 2001 was the considered gold standard. Now again, 2018 is also considered gold standard. Now ICH, it's an international conference for harmonization. GCP, ICH, GCP we say, good clinical practice. ICH, GLP we didn't have, but ICH now trying to come up with draft, already they came up for bioanalytical method validation. So we need not to worry about now any country will follow just ICH. It will become gold standard once it is there. Okay, so it's a time to smell the coffee and go ahead. Okay, so what kind of dynamism basically? So I'm sure till this point of time, you guys probably understanding. So science, when I talk about bioanalysis is a highly dynamic area in terms of science. Extended testing for selectivity and biotransformation. Now this is based on recent developments. So now we have to do conversion, back conversion, interconversion kind of experiments. There we say the drugs are not interacting in the sample when they are present. And what is happening, then how we identify? So we have protocol. We test these drugs, mass spectrometer. We put all the transitions, and then we try to see something is appearing in presence of other drug. In this channel, something is coming for that drug also. Molecular drug, molecular formula we'll check. So we check a lot of checks and done at sample preparation level, at mass level, and then we try to say no interconversion or back conversion. So we have designs. Pro-drug, drug, metabolite, metabolite to drug, drug to metabolite conversion. These kind of experiments we have to do in lab. Matrix effect, like basically in presence of when you have liquid and you inject on MS, and when you have injection and you are getting signal, when you have plasma and you are getting signal. So in presence of plasma components, either your signal on MS will increase or decrease. That is called matrix effect. Matrix means plasma, sample environment, urine, plasma, blood, from where we are extracting. So components are getting extracted along with the drug, and those components may hamper or enhance the area of the drug, which is unwanted. We want to have same area, like we validated for 100 nanogram, we are getting 1000 CPS, like some unit of MS response, then we should get 1000 only. If it is happening 1200 or 1500, it is wrong. So we can get overestimation, or if it is reducing, then we can get underestimation. So we don't want that, because we are into quantitation, should be perfect. And for most of the bioanalytical area, for standards, like we have to have within 15% ka limit. So it should not more than, in any experiment, within 15%, except of LOQ. So if it is LOQ, lowest limit of quantitation, like in place of Okay, 20 nanogram is LOQ. So that 20 nanogram is also a lowest standard or lowest quality control. There we have 20% variation. That is okay. So another important thing, incurred sample reproducibility, ISR, incurred sample stability, incurred sample accuracy. These are the new things which we, incurred sample means the actual subject samples we need to use, and based on that we evaluate whether my method is working or not. So we analyze the samples once, we get concentration, profiling, everything. Then we analyze another time. So now this value 1 and 2 matching or differentiating. So we can say method was working nicely on day 1. After validation and everything I'm talking about, it's the analysis day 1, and day 15 when I run MS and everything and did process and get the results, 15 nanogram, 15.95, 15.9 something, within 15%. That should be within 20%, 2 days. Okay. So if it is happening, method is under control. If it is not happening, another day, different results, method is not under control. So we have to do the stability, as I mentioned, whole drug, incurred sample stability, these will be done using the samples. So now I was hearing a news where people are saying your thoughts and your dreams will be captured by a chip. So the world is also dynamic. Now COVID came, you know. So regulated bioanalysis, science, people say science is almost done. Means when you know chemistry, it is okay. But I say a lot of good science you need to learn to sustain in this field. When I talk about quality standard, as I mentioned, here we have chemistry because we have biological samples coming from biological origin, human. So you need biology. We have, so GCP, biology means, human means GCP, good clinical practice. Okay. Then GLP, you are having lab, you are doing experimentation, this and that, GLP. But in post analysis, when something happens during analysis, you need to do investigations, corrective actions, training, and so many things, root cause analysis, impact analysis. This is a part of GMP, post-analytical phase. So you need to understand this is hybrid science, you need hybrid quality standard. So bioanalysis basically involves human samples. Here it comes to GCP, analytical equipment and laboratory, GLP, qualification, failure investigation, root cause, impact, GMP. So which standard I should apply for bioanalysis? When I talk about dynamism, why it is dynamic, I didn't know. But then as soon as we grow, we understood the whole world, and I'm not talking about myself, I'm not talking about India. Whole world went into that dynamism. If I say 98 to 2020, still there are new things. Recently we got DI guidance from WHO, data integrity guidance. That's also happening. So many new things happening. So GCP, GLP or GMP, single, not useful because it's a hybrid science. So hence bioanalysis demands probably a very hybrid kind of quality standard, which is GCLP, good clinical laboratory practices. What is it? Scope देख लेते हैं. So it is applicable to the laboratories that analyzes samples generated during the conduct of clinical trial. Any trial which involving human and samples are coming, there it is applied. So where blood samples, if you are handling, GCLP should apply. Pharmacokinetic studies, I said, bioanalytical, bioavailability, bioequivalence studies, pharmacokinetic studies. These studies, if you are doing in your lab, then GCLP has to be applied, not GLP or GMP. Okay, analysis of ECG, like diagnostics labs. So we have different testing there also. So GCLP is concerned with producing reliable results, which are reconstructible. So it is again a kind of GLP, like good laboratory practice, but it's a good clinical laboratory. It is more relevant, filling the gap of sample management and other aspects of clinical samples. So it is more suitable. GCLP provides a bridge between GCP and GLP, which was not there. So what it does, it ensures the reliability, quality, integrity of work and results, which are GLP expectations. It also ensures proper handling of human samples, chain of custody, sample management I'm talking about. Getting the samples from clinical, storing, then again giving to analyst for sample processing in lab, then again getting back. This should be under, because we are handling now human samples, not only chemical samples. So good clinical practice has to be applied. Ethics has to be applied. Ethics are very important focus for clinical medical practices. Doctor, when you go to doctor, Anytime we raise question on doctors working, because we believe doctor is God. Why we believe? Because doctor is doing his job without anybody's supervision, following good ethics. That's why we say God. So GCP is more of ethics, GLP more of processes, GMP highly process based, but GCLP it's a mix, emotions, clinical, human, and machines everything. Now, what kind of dynamism happened in regulatory? I divide into three areas when we didn't have anything. So before 2001, in 90s, we didn't have anything. We had some white papers, workshops, reports. Based on that, we devised reproducibility, accuracy, specificity, those kind of experiments we used to do for validation also. No much guidance. Second era, I divide post-regulated regulations era. There, 2001, I say demarked area in bioanalysis where first gold standard, first regulatory guidance for bioanalysis came for validation. US FDA, May 2001. Now it is a dynamic area because we have went through a lot of challenges, changes, big work, a lot of things happened globally. So a lot of conferences happened, problems, challenges discussed, white papers came, research articles came, machines improved, nanograms, picograms in place of micrograms. We went down to now femtograms. So many development happens, so many regulations change happened, so many GLP, like when we say quality practices also changed. All these things happened. Sorry. Okay, so that is highly dynamic area. So regulatory wise, I say this is a dynamic area where we are living today. And now, very good example I would like to tell you what happened. How much dynamism. See, GCP didn't change much. If you see the GCP first guidance, maybe 20 years back, and today, not more than 12 to 18% changes you see. But if you see GLP guidance, or if you see method validation guidance for bioanalysis, bioanalytical method validation guidance, 2001, you just check, go and check, read on my recommendation. And now you read 2018. Even you read after 10 years, only 2011 EMA guidance. There is a huge change, 40 to 50% change in guidance. You understand? It's a regulation basically. It's a law of the land. 50% change in laws. It's a huge change, even in regulatory. So how can, see, when the whole world is changing, how can I be sustaining without changing? Same thing is today I am realizing when COVID came in our life, right? So anyways, so it's a highly dynamic area, and this dynamism, probably I gave some examples so you might have realized, and that's the reason we need to be continuously on toes if we want to sustain in this area as a scientist, bioanalytical scientist, or even the lab wants to sustain, they have to be really progressive in thinking. So now the part one, second thing, what are the challenges for existing lab? And if you want to go for a good lab in future. So these are the few challenges I say very quickly I can go through, and then we can move to the second part. So first challenge, I say the repeat analysis. People, this is still a gray area, PK repeats especially. Data integration, when you integrate the chromatogram, when you get the spectra, when you get the chromatogram, basically how you are integrating. Data integrity is a different area altogether, big challenge. Still people are learning. As I mentioned recently, we got some draft guidance from WHO. Training is evergreen area, like repeat analysis. Training is very evergreen area. I'll tell you, I'll give some examples. There are a lot of mistakes people does, and still trouble. Outstanding regulatory findings. So some things which are coming even today after inspection, these are still area of improvement. So first thing, repeat analysis. So challenge, and then I'm sharing how we handle also very quickly. So most of the time people find they repeat the sample, they do the repeat analysis, but without rational, without reason. Reason has to be there. And for that reason, withdrawing that reason to guide for this reason, a newcomer, suppose I am trained, a new guy is coming and he wants to do a repeat analysis. We have to have SOP in place, good standard operating procedure. What other things should be there? We need to know the documentation part. We should document initial and repeat both the values nicely, clearly, no cutting, nothing. Reported results and online in time with the time and signature. You record everything with the time and signature. So we design different forms there we fill while doing the activity in lab. Reported results properly, assay, run identification, properly identified, run, time, date, everything, equipment is identified, calibrated. Reason for repeat analysis clearly you mentioned. Why repeating? There is a poor chromatography, there is a loss of samples, it seems half of the area something you are getting, so reason has to be there. Next, authorization of the reanalysis. So this analysis, if we are going before do any experiment of repeat analysis, the HOD or the senior person, senior group leader or HOD has to, especially HOD has to approve this, because he should know something is getting repeated. It's a critical thing. Regulatory people will come and ask, and they may come after 5 years. So how you remember 5 years down the line? So you have to have proper documentation. So in regulated bioanalysis, equal weightage has to be given to science and documentation. And we teach people like that. So regulated bioanalysis is different in two terms. First thing, we have biological samples coming from human origin. Second thing, documentation. Somewhere in standard analytical lab, it's okay to escape some of the steps, but here, no, nothing. Repeat analysis of a clinical and preclinical samples should be performed only under a predefined SOP. So we have to have SOP in place so that we can avoid all these issues. Otherwise, they keep asking, and after some years, even somebody will not remember why we did that. Even then we remember, it is difficult to identify the reason. So it should be done properly. SOP of guideline for repeat analysis, reason for repeating, and acceptance criteria should be there. So SOP should have clearly why we are repeating, when you repeat, which kind of approach you adopt, investigate how you do, acceptance criteria, when you accept, when you not accept. It's a typical profile. Otherwise it should be red means should go ideally, but black means this is happened. One sample came high. Now what to do? So repeat analysis has to be done. So SOP has to be done. Now you do reason for repeat. So repeat analysis for clinical samples for regulatory purposes. Sometimes regulatory will ask, who repeat this? Why didn't you repeat? We got good results, accepted, we submitted, but they observe, they say, no, repeat this sample. So we repeat that, that may be one reason. We repeat inconsistent replicate analysis. We repeat earlier analysis and repeat analysis, we have different values. So again, we go for repeat. That is also another reason. Sample outside the assay range. So we have 100 to 10 nanogram to 100 nanogram calibration range. This is my range, concentration range, but sample is coming 120 nanogram higher. So we need to dilute and we have to take it in that range, and then we anticipate it should be within the calibration range. That kind of assays we need to do. Equipment failure, processing errors, poor chromatography, this peak is not proper for that sample, we can go for repeat. Inconsistent PK data. This is very gray area. That's why I'm saying repeat analysis for PK reason. PK data is very gray. It's you need a lot of expertise to identify and handle this kind of samples, because whatever you do, your approach will be challenged by regulatory, because they doubt you did this to correct your result and pass your study. Possibly with this result, black one, your study may fail because of variation, because of this one subject. But if you do like red one, if you get your sample somewhere here, it may pass. So there is a lot of questions. How it happened, why first time it is wrong, why second time it is right, you did some manipulation, all these things. So it is very gray area. So ideally, repeat analysis must be avoided. So what I said, development. You have to have proper time spent in development and validation, and validation do properly with the guidance, latest updated experiments. And then you try to run your method with the skilled, trained people. Trained, I say, training is important, so that less error free. We have done a lot of studies without any error. We have qualified a lot of inspections without any critical findings. It is possible to do the good job honestly and recognized at international level. You need a lot of hard work learning for that. That is what happening. Anyways, next. So data integration. Next point. So when we have chromatogram, we integrate that. that we have to have SOP in place, like as I mentioned. And if you go for reintegration, there should be SOP why you are doing reintegration, initial data and second data, reintegrated data, we have to put there what was initially. So there is not proper, because of batch parameters, it is not taking peak properly. So I'm doing manually or something like, or changing that particular chromatogram. We do even the processing of data as a batch. So we set the parameter for batch, 100 samples will be processed in a batch by software. If we do one not coming okay, one peak, again we are changing the parameters. Then they ask questions. So we have to have reason, recording properly, SOP, why we are doing, right? And original data and reintegration data should be reported. Data integrity is a big area. I'll not go much into this, but I have to go through some slides. It's really big area. So data integrity, dynamism. Okay, so data integrity basically what? Data quality, confirmatory, consistency, data reliability, no duplication. This is the data quality. Data integrity is the degree to which data are complete, consistent, accurate, trustworthy, and reliable. Data governance measures should also ensure that data are complete, consistent, and enduring, available throughout the life cycle. So I am going to talk about data integrity because we find a lot of difficulty after so many years. Actually, we were never trained on data integrity. If you do the job rightly, there is no issue. But because of pressure, big labs, a lot of manual interventions, say one person, one project, 20 people are working just to have expedited analysis. So issues are coming. Dynamism of bioanalysis makes data highly dynamic. Now in bioanalysis, regulated bioanalysis, basically we have a lot of data. So data is also dynamic because things are changing. Means regulations also changing. Okay. So data integrity importance. Data integrity issues importance. If I talk about only one thing which I want to mention to all of you, if we are sensitive enough about the regulated bioanalysis, it's a very sensitive area. If any issue happens related to data integrity to this area, basically it is not only impacting the business of the that lab or company, it is impacting all the employees and country also. It's very serious. We have to do our things. It's a legal obligation now to report accurate and correct data. So it is not like my choice. I do good job or not do GLP, non-GLP, GLP options we don't have. If you want to do bioanalysis, we have to follow this. Data integrity issue break trust on the organization and all these things. So it's a serious thing. So when I'm doing, I should do my job properly, whether I'm manager, analyst, or custodian. Now this is not a very old story. This is the 27th July 2020. I'm not mentioning the name of the company and other things, probably you'll not able to see that, but this has happened recently, and some of the molecules now, drugs are banned in Europe. This CRO, one CRO of India conducted some studies. These studies are failed in evaluation after inspection. They couldn't defend their queries. And now the medicines which launched after these studies, those are banned. So you can understand now the whole market products they brought back, is suspended. So you can see the impact. And the image, Indian CRO did wrong. It's a big thing. Maybe one or two guys might have done something wrong unintentionally, but this is the impact. Big. So what we do, data integrity, to understand data integrity, to follow properly, we have guidance available, ALCOA guidance we say. So it's like the data has to be attributable, legible, contemporaneous, original, and accurate. This guidance. So as per how you do this, we have guidance. We do everything correctly. I mentioned whatever I'm telling you, if you do correctly, we are following ALCOA. We never learned about ALCOA, but we never faced any problem. So this is what they now, ethics and everything they mentioned in writing in this guidance. ALCOA plus is something what is being now followed, not only ALCOA. So Alcoa Plus, 4 more components they added: enduring, complete, consistent, and available. This happened in last 2-3 years. Earlier, 3 years back, Alcoa from WHO, now Alcoa Plus came from MHRA, Europe. Okay, let's move quickly. So 2 new concepts which I presented a few days back to Agilent International Webinar Symposium, you say. Globally, we launched that. That time I proposed 2 things which I am proposing and which probably our brainchild. We invented this almost, you say. So data integrity, these are the new concepts. Data integrity issue avoidance strategy, we say DIAS. These things, if we follow, understand and follow, we can avoid bad remarks from agencies. Another important thing is meta-analysis, which is being taught by US FDA to me through some webinar. So I am officially sharing with you guys these 2 things. If you understand and apply, these are the new concepts which will be helpful to avoid data integrity issues. Alcoa Plus obviously is a guidance. These 3 things, if we take care, will not face problems. Sorry. So, okay, next. So what is data integrity issue avoidance strategy, DIAS? We have to have 2 things. I should divide into 2 parts. Mitigation strategy, basically. So it's a mindset. Mindset means when you are reviewing our own data, when I am reviewing my own data, I have to think like a FDA inspector. I cannot do like, okay, fine, you are my friend, I can do lightly. No, you have to behave and review the data as an auditor. Does it look right? Does the conduct and data support the overall package? So data is there. Are you able to reconstruct the study analysis like it is happening in front of you, or there is some link, missing links? Those things you have to check. Ultimately, when I talk about mindset, thinking has to be like auditor. Another thing, mindset of proper conduct and data handling. It's a part of culture. So we have to have culture like that in our lab, where people should think right only. How to handle, how to conduct the experiment, how to handle the data correctly, ethics. Everything, everyone is important and responsible. So nothing is big or small. I am a custodian, sir. What is my job? Nothing. No. If I am a management heading the department, you are a custodian. I have my job, you have your job. Everybody has to do his job properly. Nothing is big or small. Because your job or my job, if I don't do properly, it may cause problem not only to our lab, to the country, to the region. So organizational culture is important when we talk about mindset. That is where we need to train guys, and these trainings, these things cannot be done if something is not done earlier properly in the beginning of the lab when it is getting constructed or building. Then you need a rigorous training for all these things. Okay, generic plan. Good system you have to have in place. Security controls. People should go through their login. Nobody should use common password. Is a good documentation, good data management, and any scope for removing manual intervention can be automate. So second part of my presentation will talk about that. Keep record every single injection, everything, whatever is happening, record it. Don't cut the dates. Single line, new date. That's all. That is the way we do GLP, the documentation. Protect data, ensure proper locations, proper server, validated servers, computers, computer system validation, everything should be in place. Periodic review of the computer system, acquire data, SOP to ensure data quality. This is very important thing. We need to devise not only conducting, reporting things in our SOPs. We need to devise proper review process as a part of SOP to review our equipments, analytical equipments, computer system, data, SOPs, data quality. We have to have periodic review within the department to save ourselves, to make updated, to save ourselves. Even after 5 years, somebody will come, we are ready. Or tomorrow somebody comes, we are ready. That is what. Train employees on proper, so in time, that is important. train employees on proper data handling, as everyone is responsible for data integrity. Periodic training, awareness session on SOP and DI guide. So data integrity is latest, and lot of concerns are coming related to data integrity. That's why I'm saying data integrity is very hot topic. And there we need to have proper training and awareness sessions regularly for not only data integrity guidance, method validation guidance, for SOPs what we have in place, and SOPs are not correct, then we need to update those SOPs, and then we need to train. Data audit as an internal process, or you can do the third party, you can allow third party to do for yourself. That is also possible. So preparatory inspections should be a part of your processes. There it should be presented. The entire audit has to be presented to staff like your actual regular inspection is happening. So as a third party regulator, I go so many times, I do that. I'm a consultant. After 16 years of experience, I started my own company. So now I'm a biopharma consultant, helping so many guys, so many labs in India and outside country. So we go there and I pick up a result and trace from like procuring that drug, making that solution data records, to batch run, to report, to everything, then interviewing people who were involved in batch processing, who were involved in machine operation. I ask then HOD where they did repeat, if they have any investigation, all those things I check. Single batch, end to end, including notebook. If somebody developed the method, I ask them from where you get the literature. If you get PK value, from where? So that is what the way you should do, either within the organization through your QM, BQM, bioanalytical quality monitoring, or QA, or third party. So do the best. Verify the data integrity, audit trails, demonstrate all data is readily retrievable. Now, the good thing, understand, everything done nicely, recorded, everything done in time. We are happy. Everybody is happy after our quality conduct of that study. Even validation is good, analysis is good, everything is good. But your data storage archival is not proper. And when somebody is asking, okay, give me this method validation, you archived. After 2 years, somebody came and give me archive that method validation of that number, so and so. And you are taking 2 hours to remove that file. What does it mean? It may not be intentional, but then you are not efficient their part. So your retrieval, restorage, data, sample, everything should be efficient. That is what is required and consistent. Data analysis, this I learned, this is another big concept. But DIAS is something which is done by us in India and across the globe. So nobody thought of that. So DIAS is a meta-analysis is a part of DIAS, which I got from US FDA, attending one seminar. So meta-analysis means it's a trending basically. So you do a lot of studies in lab, lot of analysis, lot of validation. You try to see maybe 10 method validation and try to see what kind of challenges you are getting, failures, out of criteria, repeat analysis. Like if you're doing analysis, there is an error in processing, there error on machine, mobile phase, or there is a failure investigation. What kind of investigations, training issues or processing issues or what? So you try to record multiple errors, multiple things, multiple methods, and try to see the trend, which are coming more. Repeat analysis is a challenge, training is a challenge, error on machine, error in processing, what kind of thing? You do Excel, do find out percentage. Suppose 100 challenges, 100 errors, and 25 errors are related to only sample processing error. Means you should more focus on training of sample processing people. So trending, it is basically meta-analysis. And now FDA, as per recent concept, they are asking you do this compulsorily before we announce our dates to come to your facility. You should be officially ready with that report. They never asked this. Earlier, people were doing as a good practice. Training and QA department used to keep that, but they didn't do that. Not as a regulatory practice. It's a good quality practice, good practice I say, but now it is a regulation. So they are asking for that. And they are saying either you need to submit remotely, or we can ask, then you submit, or we can come, then you show. But do this. So meta-analysis is basically you store the data, then there is a metadata generated. Based on that, you can sort. If you know computer, sorting the data, you can sort errors, you can sort the data, different studies, this and that. So that is from they got it. So meta-analysis is a very important part to maintain the quality, to avoid these kind of issues. Training is evergreen. Till the time we are in lab is having automation, lot of machines, this and that, but still for taking decision. Now people are saying artificial intelligence, but still drug discovery needs, even in this era, a lot of human brain to take the decision. So that is what I need. So manual intervention, whenever it comes, there are chances of error because we are not machine. We cannot be consistent as machine because we have emotions. Today my mood is not okay. Somebody came to me and say, sir, kya karna hai iska? What kind of decision? Yes or no? P ke lena hai, nahi lena hai? All those things they say. I say, okay, lena. And if I am not in mood, not emotions are there, it may be wrong decision. So I need training. And same thing I need to do for others. So when there is a human, there are chances of error. When there is a human, there are chances of inconsistency. When there are manual intervention, inconsistency may be possible. So we need a lot of training. Training of mindset, training of technicality, training of quality, training of regulations, training of balancing, and then training of consistency. Then training of SOPs, preparing good SOPs, preparing procedures so that we can get consistent outcome each and every time. Training is important for consistency, consistent outcome. And most of the audits, today if I talk about problems, data integrity and trainings. Data integrity, errors, and investigations, corrective actions. Training, anything. Who the guys who has done identify the samples not properly identified, not training properly identified. So something happened human. So training is evergreen area. So we have to have proper training. Their JDs, job description and CVs should be in place, and proper SOP for training, detailed SOP for training. Trainings can be on SOP, trainings can be on processes, training can be hands-on, training can be within organization, training can be from the conferences outside. They can call the trainers from outside. I'm WHO trainer, I'm going, so people are taking help. So training records are to be maintained properly. Again, everything is done nicely, but training records are not maintained properly, then again you are gone. Now somebody is coming and checking, it is gone. It's not good. Okay, great. Now another thing. So outstanding findings. What kind of findings we are getting now, even after having all these things? This is again part of dynamism. Challenges. So poor understanding of quality and regulatory requirements. So people say, sir, we know this guidance, this is saying this, but their understanding on that, sometimes they say between the lines, we couldn't understand. So quality control samples, suppose we have 3 concentrations, 3 samples per concentration. So they say 4 samples, sorry, 3 concentration levels and 4 samples, so 12 samples. So 3 levels, 4 samples. Now they say total 67% samples has to be passed, and each level 1, each level 1. But in that case, in guidance, they have given 2 samples, 2 samples per level. Total 67, each level, only one sample has to be passed. Now people started saying, even then they were taking 4 samples per level, and they're saying my 1 sample is passed now as per guidance. But from day one, I followed that. If I'm taking 6 samples, I say, boss, minimum 3 samples has to be passed. In my validation, I used to take care, all 6 should pass. But in worst case scenario, at least 3. If 3 are not passing, I say reject the batch, go again for analysis. Rerun, reprocess, rerun. So understanding what they are saying and then implementing properly. So poor understanding of quality practices and regulatory requirements is one of the biggest area. There people are saying this reason of repeat is not okay. This investigation is not okay. Corrective action is not devised properly, completely. It is not complete. Poor implementation of well-written achhi SOP. You got somewhere some good references or through your friends or companies, got a good SOP, but when you implement, people are not able to do that as per that SOP. So good SOP doesn't mean anything. Good SOP means the SOP you have written cautiously, good SOP, and then you are able to implement and follow. If you are not able to follow good SOP, it is bad SOP for you. It's SOP deviation. That is again a crime in terms of regulatory. Repeats. When and why? How? So when you say repeat, it's a very critical area. Repeat analysis has to be done properly. When you need to do, should define in SOP. Why you are doing, reason has to be very clear. How you do, 2 samples, 3 samples, different days, these many samples, this is the way, everything has to be defined. SOP wise, in SOP, and then method wise. Then you go, then you record. If anything happens, again record, close it. Take multiple departments help if it is not. Go to formulation, try to discuss with them also. This happened, you know about your molecule more. So tell me, is there any interaction, any other chemical which may be involved minorly? You didn't mention in content, but you know, small amount. Yeah, I added TFA, I added for that. Then you can say, oh, on my MS, that may be a cause. I don't know. So those are things. Anyways, documentation, neglected area, because scientists like science. When you say document, they say, oh, no, sir, I am not feeling good. I say, if you want to sustain, if you want to grow, you need to learn documentation. Documentation cannot be missed in regulated area, regulated lab. Investigations, root cause, these are the missing areas. Root cause not proper, impact analysis missed, kappa is not, pathetic approaches incomplete, major concerns. And training, insufficiently trained staff. Lot of pressures to perform, limited resources, less budget, more profit, less manpower, more work. And people want to go on time. People are not like earlier. People are giving hard work. Even after 6 years, 6 o'clock, they are working 12 o'clock in night and they are not crying. But today's generation, you know, they need to do so many other things. So they may go on time, but if they are going on time, then we are compromising on quality. So properly trained, mentally, physically, scientifically trained guys, and then they should understand their job. Every job is important, nothing big or small. Insufficiently trained is the major maximum findings we are finding in this area. It's a very important area. So coming to almost the end of this first part, where I was talking about dynamism, we discussed, or I talked about, or we understood about the bioanalysis, regulated bioanalysis, bioanalytical lab, okay, and so many other things. But then the dynamism and why it is dynamic, and then how it may impact our life or bioanalytical lab's life. Then we talked about even challenges. Now I'm coming almost the last part of this, last slide of this part, where I'm talking, raising some questions, so we can move to second part nicely and we can start with a big bang, right way. So what is green? Quality science with ethics. This is what we need to understand if you want to go ahead with the right mindset for the lab. In my average lab, I need to convert into green lab. Green means the best identical lab for me, ideal lab for me. So quality science you need to understand. With the ethics we need to perform. Alert and ever ready. Today, if somebody is coming, I am performing, and somebody is coming to check my job, I should be ready with the documentation and science and everything for audit. Isko kehte hain audit ready. Gaps are still here. Training and kappa, as I mentioned last slide only, training and corrective actions, investigations. Those are the areas where we need to develop. We need to be trained ourselves. We need to be alert. So are we ready to change and adapt? Adaptive to current and futuristic changes and challenges. We'll talk about this in next part. So we have to be adaptive. If we are understanding this part, then we are ready for green lab. Green lab is really going to be very good. I'll show you. Innovative as solution to challenges. Innovative as solution. So challenges will come when you work. Mistake may happen. Challenges will come, but then are you innovative enough to find the solutions? Mentally, physically trained, training wise. Okay. So paperless environment is what is the target. So we have to be automated as much as possible. Paperless, is it possible? It is possible. That is what we are going to talk in next part. Ultimately, fit for purpose. A lab has to be fit for purpose. For what we are intended, that outcome has to come out in time. That is also with the quality in time. Outcome, scientific, regulated, in time, proper output in time. That is important. Okay, after time, it will not make much sense. So when I talk about green lab, which is basically fit is hit. In Hindi we say fit hai to hit hai, right?